This recorded session is dedicated to part 2 of our introductory module on parasitology. So we already covered half of the terminologies, concepts that are important for us to grasp before we dive into specific groups of medically significant parasites. So let's begin by looking into how the parasite causes harm or injury to the host. So there are three things that I want you guys to remember. One, similar with medically significant bacteria, multicellular and unicellular parasites are able to express enzymes that can damage tissue, enzymes that can break down macromolecules, directly affecting host cells and host tissue material, lipases, proteases, nucleases. So those enzymes that can break down macromolecules once expressed by the parasite can affect the integrity of host tissue, host cells. Also, um, that beginning point, that start point relates with destruction and invasion of host tissue. So the enzymes that are expressed by parasites once secreted in the ambient or the locality where they try to establish themselves, these enzymes will break down the integrity or will affect the integrity of host tissue. As parasites will require permanent adherence, no? In most cases of medically significant human parasites, they will require a state of permanence or a period of permanence to their host tissue, to the host organ or cell. No, the enzymes are responsible for the eventual destruction of host tissue. And lastly, parasites can deprive the host of essential nutrients and substances, as they might also require these nutrients. No, they will try to sequester those nutrients, and of course, that the ultimate effect of that is the nutrients become unavailable for the host to absorb. Okay. There are several factors that affect this interaction or period of infection that parasites um, try to achieve once inside or once on the host. And these relate to factors we are already aware of, similar with how bacterial and fungal pathogens affect their host or affect the human body during their period of infection. No? Primarily the genetic makeup of the host, as there are parasites that will find it hard to establish themselves inside the host because of the given genetics that make them incapable of doing their or ensuring that infection is going to proceed successfully. No? A good example of this would be um, a human population or ethnicities that have innate or natural immunity to plasmodial parasites, primarily attributed to the kind of red blood cell, the morphology of red blood cells that they have. There's specific ethnicities that are naturally immune to malarial parasites because their red blood cells will not allow entry and invasion by malarial parasites. And that has something to do with the genetics of that specific group. Another is the nutritional status of the host. So there's this um, idea that parasites have the innate capacity to sense whether their host is nutrient rich or not. No, and in the case of a parasite that is trying to establish itself in a host that is not healthy, no, the nutritional status is said to be low, said to be subprime or suboptimal. Okay, period of infection or illness might be shorter because the parasite will not be able to get what it needs from the host, the nutrients that it needs from the host. And most importantly, setting, factoring in the environment. No, we have immunity of the host, since parasites have been living with us for the longest time. So our bodies, our race, our species have developed specific immunity to certain parasites. Programs, public health programs, vaccination, deworming, um, what else? So other examples that may relate to immune enhancement, other specific interventions that relate to immune enhancement. Okay, so the overall immunity of the host brought about by all other factors considered, genetic and environmental or external. Okay, we relate the overall immunity of the host with the period of infection, how short or long it will be. More immunocompetent individuals will have a better response to, or better capacity to eliminate the parasites as soon as possible. Okay? There are possible outcomes of parasite host interactions, whether possible that the parasite will fail to establish itself in the host, eventually it dies. The parasite is able to establish itself, but the host is responsive, immune competent enough to eliminate infection. The parasite becomes established. The host begins to overcome the infection, but is not totally successful, or there's partial elimination of the disease causing parasite. A potential outcome is also when the parasite establishes itself in the host, and in trying to eliminate the organism, the host becomes damaged. For the case of host humans, where instead of being able to use previous immune factors to counter the presence of the parasite, no? Those previous immune factors actually affect survivability of the parasite, or even to some extent can enhance parasite survivability. Good example of this is the phenomenon of antibody dependent enhancement. Some parasites may have the capacity to make use of antigenic variability. Hence, once a secondary or tertiary infection happens, even though the host already has specific neutralizing antibodies, or antibodies that are specific to the initial strain or initial antigenic variant of the parasite, succeeding infections may not be resolved, because instead of the initial antibodies also attacking or neutralizing secondary, tertiary infections, might not be the case. No? And ultimately, parasite establishing inside the host and being able to kill the host. That's about the most morbid, mortal outcome of parasitic infections. Okay. Now, so I'll talk about the physical barriers that are present, preventing parasites from uh, um, uh, preventing parasites from a successful initial attempt, no, to establish themselves or to gain entry rather inside the host body. So these first line of defense against pathogenic parasites are the external parts of the animal body, the external openings of the host, like mucous membranes, openings that can be created through the skin, like the case of intact skin being destabilized by parasites capable of burrowing into intact skin. Components of body fluids, immune factors that are present in bodily fluids are important aspects of defense, as they can serve as initial material or initial factors to control successful parasite adherence and parasite invasion. No? Antibodies that are present in bodily fluids are good examples of these components that can serve as first line of defense. Also, there are some non-specific factors, inflammatory, phagocytic, that can lead to um, unsuccessful unsuccessful host entry on the part of the parasite, of the invading parasite. And then, of course, some physiologic functions of the body. A good example is coughing, okay? For the case of parasites that can lodge or prefer the respiratory system, yeah, coughing can serve as a reflex to eject parasites that inhabit the respiratory areas, the respiratory tissues and organs. Another would be, of course, the reflex on, or our they call this normal our normal bowel movement. Okay, so let's say the host unknowingly ingests parasite laden food or water. So normal bowel movement can eject these parasites, no? The eggs or the cysts, for the case of helminthic parasites, the eggs can be washed down the GIT by the normal working of the gastrointestinal tract, especially the intestinal section. Also the same with protozoal parasites. Protozoal parasites that cause amoebiasis or dysentery or upsetting parasites that upset the GIT. Okay, the body can try to force them out through bowel movement. Okay, right. Now, how do we respond as hosts? How do we respond to parasite invasion? So two things in general, and I'm sure you're pretty aware of immune responses. So we have innate and acquired. Innate immune response is when the body detects and eliminates the pathogen using non-specific mechanisms. No, phagocytosis is number one example by phagocytes like macrophages and dendritic cells. So these immune cells can, once they recognize the presence of the parasite, they can ingest the parasite and kill them, no? Phagocytose them. Then we have acquired immune responses, is when the body, the human host, exposed to the parasite, develops specific immune response against it. So this relates to maturation and cascade, where T and B lymphocytes are involved. Um, specific specific effector cells that can target parasites, once that they are recognized, are developed, and they circulate, they maintain a particular pool for circulation in the human body. Okay. Of course, in return or in response, parasites will try to evade our immune attacks, or how our how our immune system will try to clear them. Parasites will have evasion mechanisms. So some parasites can be resistant to infection, allowing for survival upon entering tissue of choice. So a good example of this are helminths. They have cuticle and integument that can resist macrophage attack. They have cuticle integument that can serve as protective barrier against enzymatic degradation. Okay, so innate resistance to infection. And then we have immune suppression, when parasites can reduce immune function of macrophages. They lower macrophage activity, and they can, and in some cases, parasites that can prevent antigenic processing, no? So parasites that secrete factors, proteins that can inhibit antigenic processing for acquired immunity development. Those are two good examples of immune suppression. Another evasion mechanism that is very popular among unicellular parasites is antigenic variability or antigenic variation, no? Or some other synonymous terms, antigenic hypervariability. Okay, this is when so in general, this is defined as a phenomenon in which the parasite is able to express a different antigenic profile. Therefore, its previously expressed profile, in which the assumption is, the applicable assumption is, the host already developed specific immunity to that antigenic variant. If the parasite is able to carry out hypervariability, secreting or developing a new antigenic profile, the acquired immune profile is no longer is no longer going to be responsive, or not going to be triggering of immune effect, or of immune cascades, because essentially the parasite will be able to hide, no? In a way, it will be able to hide um, if it expresses a different profile, an antigenic profile. So some examples, we have Trypanosoma brucei, where it's able to express variations of its surface glycoprotein. No? Plasmodial parasites, which have high levels of antigenic diversity. So these two examples demonstrate the effect of antigenic hypervariability towards suppression of immune response to the in the part of the host, through a hiding mechanism, no? Then we have host mimicry. Okay, so this is when the parasite is able to sequester material that is host express, covering itself with that material, with that sequestered material. Therefore, will not be recognized by immune effectors. Examples, we have Echinococcus. Larval stages can carry blood group antigens. Therefore, once covered by our blood group antigens, our immune cells will not recognize them as foreign. Schistosoma is also an example that is an example parasite that can carry out host mimicry. They can acquire antigenic molecules from the host, and therefore they effectively hide themselves from immune effectors. Then there is also intracellular routes. So when parasites hide inside host cells, so we have Leishmania and Plasmodium as examples of obligate intracellular parasites. Okay. All right. Now, let me introduce you with the different types of medically important parasites that we will cover for the remaining weeks of the final term. So we have protozoans, which are unicellular. So these protozoans are divided into different phyla. Those that are under Sarcomastigophorans, we have the amoeba, the flagellates, especially the blood flagellates. Under phylum Ciliophora, we have ciliated protozoans, mostly enteric parasites. We have those that are not necessarily flagellated or ciliated, but are able to cause parasitic infection. We have the plasmodial parasites, the Babesia, intestinal coccidians, the tissue coccidians. Then we have microspora, which is actually now categorized under fungi. So they will also cover important microspora parasites. We have the helminths, the multicellular parasites, the metazoan parasites. So we'll cover roundworms, the nematodes, the tapeworms, the cestodes, and the flukes, the trematodes. Okay. Right. So that ends our part two for our recorded session for the introductory discussion on parasitology. So we'll cover in the next on-site lecture meeting. So we'll discuss our first group of medically significant parasites.